The present work, the use of biochar and rhamnolipids as enhancers of TPHs bioremediation processes, has been developed in the framework of Greener Project. Greener Project is an European project funded in the Horizon 2020 program, and it's a collaborative research project between universities, research institutions, and companies. The aim of the project is to develop new bioremediation strategies to enhance the remediation of soil and water polluted environments.
About materials and methods, for the incubation experiment, we use a polluted soil from a machinery park, where the total petroleum hydrocarbons concentration is 4,000 milligrams per kilo. As amendment, we use two types of apricot biochar pyrolyzed at 450 and 650 Celsius degrees at a rate of 5% and a commercial rhamnolipids at 1%. We prepare a 90 days incubation with the soil, the amendments, and an isolated microbial consortium from this soil, where we control the aeration and the humidity twice a week. We took the samples at different times for the physicochemical and microbiological analysis.
We analyzed the evolution of the total petroleum hydrocarbons after 30 and 90 days. And, for example, after 90 days, we observed that the best treatments were again those corresponding to the addition of the bioremediation treatment. And in the case of the biostimulation with biochar and rhamnolipids, the best result was shown with the biochar number 2. We also analyzed several properties of the evolution of the soil. For instance, in the case of the Olsen phosphorus, extractable phosphorus, and total extractable nitrogen, we observed an increase after the addition of the consortia, the microbial consortium. And this is correlated with the pH decrease and the availability of the nutrients. And finally, regarding the soil microbial activity, we observed that there was an increase after the addition of the first consortium, and this was not so remarkable after the second inoculation, maybe also due to some inhibitory effects.
Well, this is one of the experience with a very challenging soil polluted with hydrocarbons and heavy metals. The soil has been submitted to natural attenuation for a long time, and now the heaviest fractions of hydrocarbons are present in the soil. We isolated a consortium of bacteria from this soil, and this is part of our experience. In previous experience, we use compost of different mixtures. The results have been very good, but we try with this work to improve the life and the action of the consortium. And this is the idea to use biochars, two biochars, obtained at different temperature of pyrolysis. The idea is to immobilize the consortium and to increase the efficiency of this consortium. We use always also rhamnolipids as a biosurfactant to improve the accessibility of the hydrocarbons or the microorganisms. The results are still ongoing because we increase the degradation of the hydrocarbons, but not enough. We try now to use a combination of the consortium with macroalgae as a way to reach a better